Thank you, Alex, and thank you, Jean, for the kind invitation to be here today. This is a subject that may not be very popular among people who are working on single-cell genomics or proteomics or transcriptomics, but we also work on different areas, and we are really interested in seeing how well AI is performing in the structural biology field, particularly given that they were awarded half the Nobel Prize last year. So let's get started. So we always associate odor with something lovely, fragrant, flowers. So I put that thing on. But what I'd like to say is that we also have odor working.
Is that working?
Okay. Working on insects. Why insects? We'll come to that later as we go along. To get started, this is a quick overview. We will look at protein structure prediction, which has been a field of intense research since the 80s, long before you were born. Okay, so it's still ongoing. And we thought it was solved, but it looks like it's still a challenge. So we're going to look at AlphaFold, which everybody has heard of, surely, unless you don't see the news anymore or listen to all the tracks. We look at receptors for odor recognition. We have 400 genes for these in the humans, which is a very small number because animals and insects have many more. Okay, then we look at insect odorant receptors, what's so peculiar about them, and we'll look at how prediction methods such as AlphaFold compare with traditional long knowledge-based methods that we usually use. And are we there, and how we can actually get to an accurate protein prediction model. And if you're a bit afraid that this is a new area, there's plenty of stuff on the internet. You can email me and I'll send you all the details. I actually ran a tutorial on protein structure modeling for ISMB in 2005, which was judged the best tutorial. And since then, I was tutorial chair for the next 5 years. I'm tutorial chair again this year, co-chair. But these are all things that people work on. Some people do. So why? Why do we need structure? Proteins, as you know, are the worker bees in a cell. They actually do the jobs. The DNA is like the queen bee. It's got the information. The RNA could be like the drones. It conveys the message. But the worker bees are the proteins. And unlike the bee colony, they're all different from one from the other. And they each have a specific function, which they perform when they adopt a three-dimensional structure. So I loved 3D geometry. I used to do 3D trig and all the subjects. So I'm just fond of this myself. So which is why I looked at it. When a protein is folded, it looks like this is called the cartoon structure. It actually performs its function. But when it is denatured or folded out, unfolded, it doesn't do anything. It just sits there. It's called an addled egg. Okay, and this was realized in 1950. And Finson worked on it for many years. He was really very lucky. He chose a protein that can be denatured and renatured really efficiently, and it carries out its function. Since then, people have been looking for this Harry Potter style wand that can come out with a folded protein, given the sequence. The sequence is an amino acid code, 20 characters, not your DNA code, which is 4 characters. So we want to know how this makes this structure, why it forms, and once it forms, what would be the function. This is very important, especially in the genomic context. When we are genome sequencing thousands of organisms, we want to know what their proteins do. So these are all pictures I garnered from the internet because there's no need for me to waste my life making these pictures. The best method available is if your protein matches one that we already know the structure for or is similar to it, Boy, you're in business. You can quickly make a comparative or homology or what is now called TBM, template-based modeling. You get excellent results. And you can also get remote homologs by using even BLAST, multiple successive rounds of 5 BLAST. It will give you a PDB structure eventually. And even if you find that they are not easy to locate, if you can get them together, you will find that structure is conserved far more than sequence naturally, because the functional part or the business end of a protein is only a handful of amino acids, not H300 or 400 amino acids, but just 5 or 6, which have to be in the correct 3D geometry to perform the function. So it is the localization in space. So it's very much like the spatial genomics everybody is talking about, but it's at the protein level. Okay, so we are looking for this magic wand, and this seems to be one of the best solutions. You will see here there's something called multiple sequence alignment. If you have done any bioinformatics, you would have come across this. I'm pointing this out because we're going to look at this again later on. Okay, what if you can't do that? You do what is called threading or 1D to 3D mapping. Why do we do this? Biology has found that structures are more conserved than sequence, and also totally alien sequences seem to fold similarly, adopt similar architecture. Very much like our unit buildings, the skyrise that you see around Sydney, a lot of high rises, they all look similar. The insides may be different, they may function differently, or they may be office blocks or universities, as well as residential quarters. So we could have different orientations of the same thing. All we need is a scaffold on which we can drape these sequences and check their compatibility. That is called threading. That was very popular in 90s, early 90s, became very famous, and all those people are doing very well now. And it's usually domain-based. Domain meaning an independent moiety of the structure. Okay. Next case, if all that also fails, you do what is called a fragment approach. This has been the forerunner of our AlphaFold. So we have little bits of protein. Are they matching to something that is known? You collect all those spatial fragments and try to build something that looks like a protein. Sometimes it doesn't look like a protein, then it's called a non-physical model. So then you try to make one that looks biologically correct. So this has been used by many people as forerunners of what is called ab initio protein folding. What does ab initio mean?
From the beginning. From the beginning.
From the beginning, that means a priori, no assumptions. We did not think of anything, but you will see that is not true. That everybody has thousands of assumptions cleverly hidden under the carpet. Okay, so that's what you're going to look at. So this is a newer slide, as you can see, 2021. So you take the sequence, you look for fragments, you look for multiple sequence alignment, and then you try to see if there are interactions between them in 3D, and then you make your fold. So this has been very good for small proteins, but then suddenly it became available for big proteins. Before that, let's look at accuracy. There's a guy called Yang Zhang who used to be in Michigan. He's currently moved to Singapore. He's done enormous work in this area. So this slide is from him. And why do we look at this? Because the end result is you need a structure that will actually help you to do something with it. And what is it that you want to do? We are looking usually for small molecules, metabolites that perform the function. They are either signaling molecules or they elicit some kind of reaction. So to get this element organized, we need to have very accurate structural models. We can solve the structure experimentally, but it takes a very long time. There are people in structural biologists in University of Sydney who do that still, but it's a very complicated task, time consuming, and it's very expensive. So in order to just get a quick structure, we need a good model, because without that, we cannot do ligand, drug, or inhibitor or agonist design, because it's the small molecules that carry the value. Okay, till date prediction methods cannot help us with functional details, especially because if you do a mutation and the function is lost, can we show that computationally? So that is one of our biggest challenges. On the one side, we have what we want to do: drug design, ligand docking, mutagenesis, enzyme active sites, finding the topology or the family of an unknown protein from a genome sequencing. So for all these, you also have the methods that can help. And we are in this region, and therefore we need a good or high accuracy model. And free modeling or ab initio modeling is here. It can go up to here, but it's still not able to help us with mutagenesis. Okay, so that's our problem here. Now, protein structure prediction has been around for many years. I would consider the first structure success in this from Powling's lab. Powling, Corey, and Branson predicted the structure of an alpha helix well before anybody found it using X-ray crystallography. So that was perhaps the first element of protein structure prediction, but it's not a full protein. He only predicted an alpha helix. It's secondary structure, but that's by the way. And then we have the first homology model being built in 1969. BLAST can pick up structural homologs as early as 1990. If you run BLAST repeatedly, you can pick it up. Then we have fold recognition and threader in the 90s, followed by modeler, which is a very good program still in use for building models by André Chaly. And then 1997, we have SciBLAST for distant protein recognition. Now, where does AlphaFold comes in? It's in the next round, in the 2000s. Okay. We had here the development of folding, etc. And then 2018, we have AlphaFold being made available to the public by Google's DeepMind. They were not really doing that well, but it was seen as a very promising methodology leaning towards free modeling. Okay, at that point, they were still trying to fold a sequence from first principles. And you will see many other methods here. DI-TASR is from Yang Zhang's lab, Rosetta by Baker. Baker also got the Nobel Prize last year for protein design. So you will see a lot of big names coming up towards the end of the slide. But by 2020, AlphaFold suddenly became extremely good. Okay? And it already went to the level of experimentally determined structures, which is 90. It's coming up to about 88 here as far as accuracy goes. And therefore, it was decided that it's a fantastic method. We have what is called a critical assessment of protein structure prediction, has competition held globally every 2 years. And there it was found that it was doing fantastically well, as good as experimental structures in 2020. Lo and behold, science said this is the best breakthrough of the year. They got the Nobel Prize last year, and this was the Nature briefing. This has done a brilliant job, and they have been able to predict structures for thousands, millions of protein sequences in UniProt, GenPept, etc. Okay. The great thing is if you submit a sequence to the server, it takes about a few minutes. It's like blinking. And then you have a structure. It's all done. No hard work. Boy, isn't that good? However, they always seem to fall short of our requirements. And we'll show you why. What's odor recognition? Do you know? Do you know that we have what is called odorant receptors? You know that? How many of you know that? I know Jean Yang will know that. Maybe Alex. Okay, we have what are called odor receptors, which are actually G protein coupled receptors. Anybody heard of GPCRs? They already are so big, they had to get another acronym. Okay. We have 900 GPCR genes in our genome, 900. And about 400 of them are odorant receptors. GPCRs are the largest family of proteins that your pharmaceutical medications target. So the drug companies love them. So if you want industry-based grants, remember GPCRs. They are still funding lots and lots of grants. So this is from the mouse. We have foods. These are all the receptors that we have for smell, for foods, floral, social, and survival. Not for us. We don't have so many survival receptors, but many other creatures do. So what are these GPCRs? Ignore everything except the ones that I put up. It all started with adrenaline, also known as epinephrine, be activating receptors, adrenogenic receptors. And that was very important for heart and other diseases. So this was being investigated way back in 1905. Then a person called Gilman purified these receptors. So the G actually stands for Gilman, but very few people know that. So if you know that, you can answer a quiz question. Okay, so it's a Gilman G protein coupled receptor. He found all the G proteins. They are also named for him. Lefkowitz then named the receptor in 1980. And Kobilka, who also got the Nobel Prize for structural biology, discovered that they form 7 transmembrane bundles. So he called it the snake. So that's the critical part. And you look at the structure here, it looks something like this. It's got an N-terminal tail, extracellular, rest of it is in the membrane, and a small C-terminal tail that is intracellular, and the ligand binding site is between the membrane, transmembrane regions. So there is nothing else in this protein except the membrane part. If you do biochemistry, you'll also learn about protein folding, trafficking, how it's transported across the membrane, and there is a big chapter on GPCRs. Why GPCRs? They are the most efficient receptors. External ligands bind to them, and they are the fastest acting ligand receptors in the human body. Almost instantaneously, a message is sent from extracellular to intracellular via G proteins. That's why they are called G protein coupled receptors. The odorant receptors were found in 1999, and Linda Buck and Richard Axel got the Nobel Prize for that. And that's what we are interested in, because they are a subclass of the odorant receptors, and they actually are totally unknown. More importantly, they not only occur in your nose, they also seem to occur in all kinds of other tissue. When they were first found, they were also found on sperm cells. So what are odorant receptors doing on sperm cells? What are they smelling? I don't know. But you see, there's a lot to be known. There is at least one odorant receptor in 91% of cancer cells. So can this be used as a targeting agent? So that's what we were interested in. So we looked at some of them, which are possible biomarkers. for breast cancer, prostate cancer, colon, and small intestine carcinoma. So we were interested in that. We were working on odorant receptors first, which are 7 transmembrane protein receptors. We did work on identifying new ligands using machine learning for gastric cancer. Then we built models. Then we also identified templates because GPCRs have lots of templates or known crystal structures. And we also found ML-assisted ligands, which were actually better than anything that was ever known, verified by Matsunami in Duke University. He also then later on, about a few years later, went to solve the first experimental human odorant receptor structure. There's only one, and it just came out after we finished all this work. So it didn't help us. We then started working with teams that are working with insects. Insect odorant receptors are very important for their behavior. It helps them to find food, mates, etc. They also are a big loss to our agricultural industry. About 20% of the crops are lost due to insects. And the most famous of them is the Queensland fruit fly, Bactrocera trioni. They are very different from Drosophila, which is the vinegar fruit fly, because they branched off many years ago. But we do know that analogs of natural compounds, such as Q-lure, is very famous, is able to attract them in traps before they can attack the fruits, and therefore we are able to save our fruits. We need new ligands for pest control. This is an economic problem, and it doesn't involve any ethical concern, ethics clearance, or human subjects, or any of those problems. So we work on the insect odorant receptor and how to attract insects, keep them away from our crops. They function very differently, although they have 7 transmembrane helices. They look very similar to a GPCR, but they don't need G proteins. They work with a co-receptor, and it looks like this is a very primordial receptor that is able to bind the ligand and transmit the information intracellularly by opening up an ion channel and letting ions in. G proteins also induce ion release intracellularly, but via a very complicated mechanism. So this could have been the primordial way to do the transfer of information from extracellular to intracellular. We have 7 transmembrane helices. This is the GPCR that we looked at earlier, but the insect odorant receptor is flipped 180 degrees. It's inside out. The N terminus is inside, and the C terminus is outside, and there are big loops on the inside. So we started work only in 2022, when there were only 2 sequences with 4 experimental structures, and these were known to be homomeric, but in vitro they function as tetramers, and homotetramers, one is an orco, and the other one is an OR. There were only 2 sequences from which we had structures. Very low sequence conservation, less than 20% identity among these 2 receptors with structures, and very much less with totally alien odorant receptors. Homologs are also known in animals, gustatory receptor-like proteins, but they have not been well researched yet. This is a very novel family called the 7TM family. They are known even in human beings. We have quite a few evidences for these, but they're not well researched yet. So this new area, if you want to look at it, where do the ligands bind? They bind very similarly to a GPCR. This is the bovine rhodopsin with the retinol bound. This is an insect odorant receptor with the ligand bound. The ligand is DEET. One of the insect sprays that we use contains DEET. So this chemical is bound exactly in a similar location and the site is accessible from the extracellular side, and the transmission of information occurs intracellular. So with this in mind, we wanted to test whether fold prediction worked compared to free modeling. When we started, we had a workflow called BioGats for finding a good template for GPCRs, which we adopted for insects. We validated this with, cross-validated it with the two structures, and also looked at AF models, but we found that AF did not do very well. For example, we applied it to Drosophilus OR59B. A single amino acid change is able to destroy ligand binding. The fold from AlphaFold, which was AlphaFold3 at that stage, is very similar. That's in yellow compared to the model in green. You can't even tell that it's very different. RMSD is a bit high, but it's okay. This is before minimization. But when we start putting in the variant, the mutation, and docking the ligand and checking things out, AlphaFold, we wanted to see whether it at least finds the functional binding site. AlphaFold declares that it can't work with variants, mutations, etc. So it's already well known. What we found to our shock was that although it bound in a similar location, DATE, which was a binder in our model, is a non-binder straight away for AF2. It doesn't bind with enough affinity. It's got a positive binding energy. That means it's not binding at all. So it doesn't help. And what happens when we do the mutation? We showed clearly that our model lost its binding capacity, became positive, but AF2 decided to bind the ligand a little bit better. So sorry, we are not getting it right yet. But this is not enough. Just, there's a saying, one sparrow doesn't a summer make. So we wanted to see extensive study. Let's take 2 pest species. We took 2 Bactrocera species, the fruit fly kind of species, Dorsalis and Mimax. 5 of this and 3 of that, and 5 small molecule compounds for which we had experimental data from our CSIRO collaborators. So we had plenty of data. So now we wanted to see whether we could use this modeling approach compared to AF2. Sorry, correction, we are now in AF3. So we wanted to check whether AF3 worked. These are the new structures that came out afterwards. So we listed those. And then post-simulation studies were done to check that the ligands were binding properly to their cognate receptors. And then the preferred template could be figured out. So we knew how to do our template-based modeling. Anyway, to do that, we needed to do a lot more work, multiple sequence alignment. We also did phylogeny and all that. I'm not showing everything. And we did heat maps. But here are the 5 small molecule ligands that we looked at. Now, what happened when we do AF3? AF3 finds the fold, but doesn't do well again with the functional data. Template-based modeling is able to discriminate between binders and non-binders, but AF3 cannot discriminate clearly between binders and non-binders. It does some, but not all. So we need to be a bit afraid of that. So we cannot get a functional model. So why is it that it's not working? So we did some backtracking and some analysis. So the reason is now everybody talks of AF. It's called the AF mania. Everything is AlphaFold. So every reviewer wants you to run multiple rounds of AF because they think it's the single silver bullet that can solve all our problems. It does provide confidence estimate, and that's according to the color. And globular folds are predicted very well. The protein structural problem has been solved predominantly for soluble globular proteins. What do we mean by globular proteins? Proteins that are compact Globular meaning they form some kind of oil drop. So they fold on top of each other. They don't like so much water, so they try to hide their hydrophobic amino acids, and therefore they form these compact structures. And there's quite a bit of evidence of that. AlphaFold was trained on that, and therefore it does very well on that. Like all machine learning methods, you give it a training set, it's going to work beautifully on a similar example as a query. When you have small proteins like insulin, anybody heard of insulin? Was the first protein to be sequenced step by Sanger. So Sanger Institute, actually he worked with proteins first. And all the things he did with proteins, he then prepared for DNA. So that's why we have the chemicals that bind to the end, etc., all done by Sanger. Okay, didn't do well for insulin, it's totally unraveled. And for some folds, it actually is called ugly by Janet Thornton, Dame Thornton, who actually is a structural biologist for many years. So there is no PDB structure to compare it with. That's not true because this is actually a fragment. That's a small part of a large protein, which is an aggregate of 20 to 30 small pieces of protein, identically folded. There are small proteins, but this is a total rip-off. It's not doing well. So AlphaFold is not doing well. Why? Why is it not doing well? That's what we wanted to know. We find that from AlphaFold 2 to AlphaFold 3, they used more materials, larger data set for training, experimental data, more multiple sequence alignments that I showed you in the initial slides, more inter-residue interactions from the different structures. So that was all thrown into this cauldron to make the magic potion, and out came AlphaFold. So it is actually threading plus fragment assembly to the power of deep neural network in my formulation. So that's formula from me, okay? So as more experimental structures come about, AlphaFold will get better. For our 7 transmembrane family, we need more training on the membrane helical bundles. We need more people to do maybe large language models, but on membrane proteins especially, and use a double model, one for the portions exposed to solvent and one for the transmembrane portions, but that's not happened yet. Perhaps we need to go back to first principles and look at true alpha folding, because another famous person, George Rose, who's been around for many years, said, deep learning represents a major advance in protein fold prediction, but this is still not the fold solution for the folding problem. So we need to learn how biology is able to do it at the ribosome when the protein is expressed without any artificial intelligence. So that's what we are not there yet. So we have done some extra work, but our paper is still in under review, so can't give you all the details. But suffice it to say, when we compiled TBM with AlphaFold3 and the new super duper lipid ligand possibility to add, we added 100 lipid molecules, enough to provide a fake bilayer around the protein, or at least the membrane, transmembrane regions of the protein. We took 4 Drosophila odorant receptors with functional mutational data, generated 3 models for each, and 2 sequences for each, one that is wild type, one that is mutant. So we took 24 of these, built the biological membrane, added the water molecules, added the salt concentration, and then sent them for molecular dynamic simulation. Takes a very long time. NCI helped us, otherwise we can't finish this job. And we calculated the free energy of binding using Poisson-Boltzmann free energy calculations after the molecular dynamics. And we can show you that all the structures a priori look similar. So at the outset, everything looks the same. So you're wondering why you're spending so much valuable time trying to do something that AI can do very efficiently. The lipid addition did not improve anything. The lipids like each other better than they like the protein. So they seem to form clusters still. This has been noted before on a blog site, but nothing was published yet. So we put all our models in print so that we have a publication for it. We did a lot of comparative analysis. And for our ligand, what we found was mutation, we lost the binding affinity, whereas for AlphaFold3, it was the other way around. The mutant bound really well, and the wild type didn't bind at all. So that was already a no-no. And lipid, both the mutant and the wild type bound the ligand. And the mutant bounded better. So there's still little more to go. These are the people who did the work. I'm only the boss. As I was telling Jean and Alex, I code in Fortran. So I'm not a very popular person in the lab. So these people write Python or all the simpler languages. So Amara did her PhD with me on the human odorant receptors. She did large-scale insect odorant modeling. She's now working as a postdoc in applied biosciences on a coral reef project, genome sequencing project. And Vanati did her MRes with me, and I am her de facto PhD supervisor for this project.